All right, we're back with our last and final video PowerPoint lecture of the semester. This is our last two chapters, amino acids, proteins, and enzymes. We begin with proteins. Proteins, biochemical molecules. They consist of what we call polypeptides, joined by peptide bonds between the amino and the carboxyl groups of amino acid residues. All right, so we've heard this term before, residues, when we talked about our carbohydrates. All right, so that just means amino acid individual pieces. Okay, so we're getting a picture of what proteins are. All right, there are many, many amino acids joined together. Proteins perform a number of vital functions within the body. Enzymes, in fact, all right, and we talked about some enzymes, carbohydrate enzymes, in our last chapter. And they're proteins, actually, that act as biochemical catalysts. So throughout our whole discussion of organic chemistry, we have, in fact, heard this term catalyst a number of times. All right, and we're going to hear it again a number of times with these particular topics. Catalysts, we saw the use of catalysts for a number of reactions. Okay, remember what catalysts do. All right, they are required for a reaction, and what they do is they lower the overall energy that it would take for the reaction to go. All right, so that maybe it goes under lower temperatures. Okay, and it increases the rate of the reaction. A reaction might take, several weeks to go, but with a catalyst, it could go in, a moment of hours. Okay, just as an example. Many proteins have structural or mechanical functions. For example, actin and myosin are proteins that have mechanical and structural functions within muscles. Proteins are also important in cell signaling, in immune responses, cell adhesion, and in the entire cell cycle. They are an absolute necessity, a necessary component in animal diets. All right, so with that introduction to what exactly proteins are, we begin with those building blocks of proteins, amino acids. We begin from the smallest unit. All proteins, as we said a couple of slides ago, they are polymers. Polymers are very long chain molecules, and they consist of much smaller units. So proteins are polymers containing chains of amino acids, which are chemically bound to one another by what is called a peptide bond. And all a peptide bond is an amide bond. And that term amide should be familiar to you, okay? You know what an amide functional group looks like. Most organisms use 20 naturally occurring amino acids in order to build proteins. The way the amino acids are joined, the linear sequence, as it is known, of amino acids that occurs in a protein is dictated by the sequence of nucleotides in an organism's genetic code. These amino acids, they are referred to as alpha amino acids. Why do we call them alpha amino acids? Well, it's because the amino group in the amino acid is attached to the very first carbon in the chain. And the first carbon is the one that is attached to the carboxyl carbon. So this is what we're talking about. So this is a general structure of an amino acid. All right, amino acid, that tells you everything about an amino acid. All right, it contains an amino group. All right, so here we see the amino group. All right, and it contains a carboxylate group, a carboxylic acid. All right, amino acid. They have an acid part and an amino part. So here's our carbonyl carbon. Okay. And the very first carbon attached to that carbonyl, right, is known as the alpha carbon. And all they're using is the Greek alphabet, and alpha is the first letter in the Greek alphabet. So they are called alpha amino acids. All right. So we have the amino group attached to that alpha carbon. All right. And in addition, we have R groups. All right. R groups, remember, are chains, carbon chains. All right. So they can vary. Okay, so we have a good idea, a good picture in our mind of the general structure of an amino acid. Let's continue. So 20, right? Naturally occurring amino acids, as we said. So what chemists do is that they classify amino acids. The amino acids are classified by that R group that we saw attached to the alpha carbon. Okay, the side chain, right? Those R groups are called side chains, and whether they are acidic or basic. All right, so they look and they distinguish the polarity of the R group and the acidity or the basicity. So it's those three criteria that are used in order to classify amino acids. All right, and that makes it a whole lot easier to remember them. All right, so again, let's refresh our memory. Okay, this is the general structure for an amino acid. Okay, and we will see why this one is slightly different than the one before very shortly. But here is our carboxylic acid part, our acid part, and here's our amino part. Okay, and so polarity. All right, so we classify our amino acids by polarity of that R group. So our first classification slot is for neutral. All right, so that R group is neutral and non-polar. Okay. Another one is neutral and polar. Our third classification group, basic, so that R group is basic and polar. And typically what that means, okay, to be in that category of basic and polar, that R group, all right, it's a carbon chain, and it has attached to it an additional amino group, and that plops it in that category of basic and polar. Okay, and then our last classification group here is the acidic and polar. All right, so what makes an amino acid fall into that category? Again, we are classifying these on the basis of this R group. All right, so something on that R group, all right, is making it acidic and also polar, and that something is a carboxylate group somewhere on that chain there. Okay, all right. All of these amino acids we're going to see as we go through them are known by not only their name, but also a 3-letter and a 1-letter abbreviation. I'm not sure if my my illustrations have the 1-letter abbreviation, but the book I know does. All right, so but they do have the name and the 3-letter designation. All right, so we begin with that first category, the non-polar amino acids. Amino acids are going to be classified as non-polar when that R group, okay, is either just hydrogen, an alkyl group, or an aromatic, all right, and nothing else. So here they are, and actually, in fact, remember we said we have 20 amino acids, right, naturally occurring, and the vast majority of them fall into this category. All right, so again, we see, here we go. Let's just point out, here's our carboxyl group, carboxylate group. There's the alpha carbon. We just have hydrogens attached. Okay, and so that's glycine, and there's the 3-letter, all right, abbreviation. Okay, and here again, alpha carbon, and we see that they're all highlighted. Those R groups are all highlighted, and in every single one of these, okay, that R group is either a hydrogen, or it is an alkyl group, or as we come down here, it's an aromatic. Okay, all right. Polar amino acids. Amino acids are classified as polar, again, based upon that R group, all right, if attached to that carbon chain, that R group, we have alcohol styles or amides. So here we have it. There are 6 of them, okay? Serine, and let's check out that alpha carbon, and here's our R group, and we see that we have hydroxyl group attached, so an alcohol. Same thing in threonine, okay? And tyrosine also. All right, we have an alcohol. We have, yes, we have an aromatic, but we've also got that alcohol. The presence of the alcohol makes it polar. All right, and we have the thiol, the amides here for the last 2 of the 6. Okay, and we move on to the last 2 categories of classification, the acidic and the basic amino acids. Amino acids are classified as acidic when the R group is a carboxylic acid, as we said. And they're classified as basic when that R group is another amine, right? So here are the last 5 of the 20 amino acids. And let's see if we could pick out who's who here. All right, let's begin over on this end. Let's find, so here's our carboxylic acid group, our carboxylate group. There's our alpha carbon. Here is the R group attached, and look what's attached to that R group, our carboxylate group right there. Same thing here. So these 2 right here are going to be classified as acidic, okay, acidic amino acids. Over here, again, let's find that alpha carbon. So here's our carboxylate group. There's the alpha carbon right here. And yes, we have an R group, but look at what's attached. It's an amine, okay? Same thing here with lysine and arginine, okay? Further down on the chain, we see an amine, okay? In addition, okay, that's an amine in addition to the one that's attached to that alpha carbon, okay? So there are our acidic amino acids, and here are our basic amino acids. All right, so 20 naturally occurring amino acids, okay, 20. 9 of those amino acids, 9 out of the 20, are not synthesized by the body. And since they're not synthesized by the body from what is there, all right, that means they're essential, okay? We need them. You've got to obtain them. And how do you obtain them? From the protein in your diet, okay? From protein in the diet. They are found in all of dairy products, so you can get them from dairy products, which is why probably from day 1 of your birth, right? Drink milk, have dairy, right? It's one way to get these 9 amino acids that are not synthesized, all right? How about, can you get them from grains and vegetables? Well, 1 or more of them is missing in grains and vegetables, all right, which makes it difficult, a little bit difficult for vegetarians and vegans, all right? They have to be very careful to make sure that they get them all in their diet. Otherwise, they'll have deficiencies, all right? So who are the 9? Well, they're histidine, isoleucine, leucine, lysine. methionine, phenylalanine, threonine, tryptophan, and valine. Okay? Complete proteins in your diet are going to contain all of the essential amino acids. But then there's also incomplete proteins. Incomplete proteins are deficient in one or more of the essential amino acids. So typically, if you're a meat eater, all right, you typically don't have too much of an issue getting the essential amino acids in your diet. Okay, so so far in our study of amino acids, you've seen that general structure drawn a couple of different ways. All right, and I said, hang on tight, we're going to get to it. We're going to see why sometimes we show that acid group, the carboxylic acid, with its acidic proton on it, and sometimes we don't. Okay, so Zwitterions, amino acids, right? As we said, they contain both an amino group and a carboxylic acid group. In our study of carboxylic acids, all right, and amines, remember what you learned, right? A carboxylic acid is an acidic functional group, and that amino group is a basic functional group. So there it is, okay? This is a general structure for an amino acid, right? And it shows the carboxylic acid part, and it is showing that acidic proton, okay? And then remember, right, amines, right, because that nitrogen has a lone pair of electrons on it, that nitrogen in that amine is basic. So you have an acidic and a basic functional group in an amino acid. Well, because they have both of those groups in the same molecule, all right, what happens when you put them in water is that you get an internal means within that molecule there, all right? You get an internal acid-base reaction occurring, all right? What does the acid do? It donates a proton. What does the base do? It accepts the proton, okay? So let's show that happening. Let's imagine that we put this amino acid into water, okay? And internally, right, it doesn't even need the water there, right? That proton is donated, okay, to that nitrogen. The nitrogen accepts it, right? What do we end up with? We end up with the conjugate base right here. This is the conjugate base of that carboxylic acid, carboxylate group, okay? And then over here, we end up with the conjugate acid of the amine, okay? And they're on the same molecule. So this, we've seen the structure written before, and that's all. When we show it like this, we're just showing what happens, all right? That internal reaction, that proton is probably not really there. It's over here, okay? And this here, because you have both charges in the same molecule, negative and positive, is known as a Zwitterion. Zwitterions are neutral, of course, because you have a +1 charge, -1. They cancel one another. So it's a neutral compound or a neutral molecule. In solution, okay, so we said this happens when we put it in solution. So in solution, the structure of an amino acid, all right, so this structure right here is what we see in solution. And if we change the pH of the solution, we can change the structure. And let's look at exactly what we're talking about there. Okay, so let's take the Zwitterion and move on with it on the next slide. Okay, so there we said in solution that amino acid is going to undergo that internal acid-base reaction, giving you a Zwitterion. If we now add acid to our solution that has this Zwitterion, okay, we're going to get another acid-base reaction. It's going to cause the Zwitterion, right, to pick up a proton, right? We have conjugate base here, conjugate acid there. We're going to add acid to it, so we're going to lower the pH. That's what that means, remember? So hydronium ion, okay? And our conjugate base will react with our acid, and lo and behold, we're going to get a different form of our amino acid. This particular one has a net positive charge to it, right? We've gotten rid of the negative charge, right? So in acidic solutions, this is the form that your amino acid is going to take on. All right, so what's our other option, right? We can increase the pH. So increasing the pH, remember, means it's basic, right? How do we do that? Hydroxide ion. We're going to get another acid-base reaction, okay? All right, who's the acidic part of this Zwitterion? Right here, folks. All right, this is our, remember we said it's our conjugate acid of our original form. Okay, so let's put base in there. Let's increase the pH. And our base is going to react with our acid right over here. Okay, and we're going to transfer a proton. Okay, right there. It's going to accept it, right? Donate, and there we have it. We have another form of our amino acid, right? It has a charge on it. What's the charge? It's negative. So this one has a net negative charge. So depending upon the pH of the solution, you're going to have a different form of your amino acid. Okay, and all these are those acid-base reactions that we talked about back in chapter 10, I think it was. All right. So what do we say about amino acids? Because they can react as an acid or a base, all right? We refer to amino acids as being amphoteric. They have the ability to act as an acid and a base. Because amino acids can react with both acid and base, if you have solutions of amino acids and proteins, right, which are made of amino acids, right, they can act as buffers. Remember the definition of a buffer, all right? So we've got both of those things right within the amino acid or the protein, right, the long chain of amino acids. For example, think of the role blood proteins play. Blood proteins help regulate the pH of the blood. Okay, if the pH of the blood becomes too acidic, that blood protein, all right, that can act as a base, helps keep the pH right there at 7.4 where it should be. All right, and vice versa. Okay. Where do we go from here? Still on our building blocks, on our amino acids. Let's move to reactions of amino acids. And the first reaction we're going to look at is peptide formation. So for this, you need to go back through your mind and recall some of our earlier discussions in our organic chemistry chapters. Recall when we studied amides, right? Amides are, in fact, formed from a reaction of an amine and a carboxylic acid. It was called an amidation reaction. All right. The carboxylic acid was combined with an amine, and what happened is a new bond formed right here with the carbonyl carbon, and we lost water, right? So we form an amide in this reaction. There's our amide, and we lose water. This was an amidation. And what resulted is what we call an amide linkage. Okay, this amide linkage, when we're talking about this happening with amino acids, we just change the name, right? It's called a peptide bond. So let's take that same reaction and let's envision it occurring between two amino acids, right? If it occurs between two amino acids, we're going to end up with a dipeptide as a product. And it's going to be held together by an amide linkage. And again, because we're talking amino acids, we then call it something different. It's called a peptide bond. Okay, so let's take glycine as an example. That's our simplest amino acid. On that alpha carbon, we have 2 hydrogens. Okay, so it's neutral. And let's take alanine. So that's our next simplest one. That our group is a methyl group. Okay. All right, so let's envision that same thing happening. So here, what do we have? We have our amino group, our amine, and here we have our carboxyl carbon. So we're going to envision this coming on over, right, and forming that amide linkage, all right? So that's all it is an amide linkage. There it is, all right? And so why aren't we calling it an amide linkage when it really is? Because that's just something that we do in amino acid chemistry and peptide chemistry, okay? We call it a peptide linkage. It's linking 2 peptides, okay, 2 amino acid units. But all it is, right, it's an amide linkage, okay? And here is our functional group, right? So there's our amide functional group, which you should recognize, okay? And again, because we've linked 2 peptides, 2 amino acids, we call it a dipeptide, all right, or a peptide linkage. All right, and of course, like we said earlier, right, we know from our reaction, our amidation, that we do end up with water. So these 2 amino acids here, okay, we joined them in this way with the carboxyl group of the glycine to the amino group of the alanine. We could flip things around, okay? We could combine the carboxyl group of the alanine with the amino group of the glycine, okay? Everybody see that, right? Because we've got one on both of them, all right? So if these 2 amino acids are connected in the other way, you're going to form a structural isomer, okay? Remember structural isomers, what they are, okay? Structural isomers, the same formula. It's going to have the same formula, right? Same molecular formula, but it just has a different arrangement of the atoms. So we end up with a structural isomer of the dipeptide. Structural isomers, remember what we said, they are going to have a unique set of physical properties, all right, which means different melting point, different boiling point, different solubilities in terms of a linkage of peptide, but they're going to have a different role, okay, entirely, okay. So here are those same 2, all right. So notice what they do. This is a glycoalanine. Notice how they put the name here, okay. The one with the carboxylate group, all right, is first. The one with the amino group comes second, all right. So who did we connect this time? Which carboxyl group? It was the alanine, so we'll call it alanyl glycine, okay, and that's how they name it. So it's also a dipeptide, but the way they're naming it is telling you which carboxyl group attached to which amino group, okay. All right, so dipeptides. Okay, well, each of those still has an amino group, and each of them still has another carboxyl group. So a third amino acid can join in the fun, right, in the chain on either end. And when that happens, we're going to end up with a tripeptide. Let's look at an example of a tripeptide, all right. So here we have alanyl glycinyl alanine. So we brought in, valine, I'm sorry, it's valine here with a different R group, okay? And again, the way that name is put together is telling you what is joined to what. And this is a tripeptide. We have 3 amino acid residues bonded together with what kind of bond? All right, this is a amide linkage right here. This is an amide linkage right here. What do we call those amide linkages when we're talking amino acids? We call them peptide bonds, okay, or a peptide linkage. Okay, well, we still have a carboxylate group here. We still have an amino group here. So a fourth amino acid could come on in, all right, either end, and that would form a tetrapeptide. A fifth would form a pentapeptide, etc., etc., etc. We can continue adding, and that's how we end up with proteins. So when you just have short chains, 2, 3, 4, 5, 6, all right, they're referred to as peptides, okay, like tetrapeptide, pentapeptide, hexapeptide, etc. Chains of up to about 50 amino acids are called polypeptides. And so then when we get more than 50 amino acids linked together with these amide bonds, these peptide linkages, then we call them proteins. Okay. The terms protein, though, keep in mind, and polypeptide are often used interchangeably. So you might call a protein a polypeptide. They are, in fact, okay? It's just a matter of numbers of amino acids linked together. Okay, so now we know how protein forms, okay? From the linkage of a number of amino acid residues. Let's move on to protein structure. The structure of proteins is a lot more complex than that of the simple organic molecules that we have looked at up to this point in time. Many protein molecules consist of a chain of amino acids. And what did we say? 50 or more, right? Okay. Twisted and folded into a complex three-dimensional structure. And that's what makes the protein. That complex 3D structure of a protein is what imparts the unique features to the protein that allows the protein to act in diverse ways. There are 4 levels of organization that we talk about when we talk about protein structure. Those 4 levels are the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. That primary structure then leads to what we see in the secondary, which leads to the tertiary, which leads to the quaternary. Okay, so we're going to start with the primary structure and see how that then cascades and leads to the next level. All right, so primary structure of proteins, what is it? The primary structure of a protein is very simply the linear sequence of side chains that are connected to the protein backbone. For instance, here we see, let's see how many peptides we have here. We have one amide linkage here, okay? We have another one here, so that's 2, right? And here is 3, so this is a tripeptide, okay? All right, so we're just showing a very short snippet of a protein, okay? And when we look at who we have here in terms of amino acids that are joined together, right? We're going to look at that R group to distinguish who exactly we have, all right, here. Okay. All right, so we look there at those R groups, and that helps us identify who we've got. We actually have 1, 2, 3, we have 4, okay? We have 4, so this is a tetrapeptide. All right, so who is joined together here? Again, we go to those R groups and we see we have alanine, leucine, cysteine, and methionine. All right, there we go. All right, those are based upon those R groups. Okay, so the primary structure of protein is the linear sequence. So what sequence do we have, right, from left to right, right, and of the side chains that are connected to the backbone. All right, so that's what we're looking at. We looked at those side chains to identify what amino acids we had here. And this is how we write it. Sometimes they have the dashes in them, sometimes not. Sometimes they're just all joined together. Okay, so that is the primary structure of this particular protein. Okay, each protein has a unique sequence of amino acid residues. All right, and that sequence of amino acid residues then causes it to fold into a distinctive shape. And it's that distinctive shape that allows the protein to function properly. Okay, remember, we draw them like this. Okay, we draw these organic structures like this, but there's those 3 dimensions, right? Remember carbon with 4 bonds, right? 109.5 degree bond angles. All right, tetrahedral geometry, as we said. So there is geometry to this. All right. Okay, so what happens then? All right. Well, that primary structure, right, that sequence of amino acids leads to the secondary structure. And one secondary structure that we see is the alpha helix. Hydrogen bonding causes the protein chains to fold and align to produce orderly patterns called secondary structures. Here's an example of what we mean by that. We see here over on the far left, okay, a model of a protein, okay, a snippet of a protein with the carbon, the oxygen, the nitrogen, the R groups, the hydrogens, okay, and it had a primary structure, all right, so how were those amino acids linked together, okay? And over on the right, we have it simplified. Okay, so it's a little bit easier to see. So we see the primary structure, okay, the linkage of each of those amino acid residues gives you a long chain of carbons, okay, with nitrogens, all right, carbonyls and amino groups. That's what it gives us, with R groups, of course, hanging off of it, okay. But hydrogen bonds, remember how they form, okay? A hydrogen bonded to a nitrogen is capable of hydrogen bonding. So those hydrogens on the amino groups will hydrogen bond with the carbonyl. All right, carbonyl, oxygen. And so when that happens, it takes that long chain that we drew on the previous slide, and notice how it is folding it, okay, and spiraling it. It's the hydrogen bonding that causes that change in the structure, and that's called a secondary structure. The alpha helix is a type of secondary structure. Is a single protein chain. That's what we're seeing here, single protein chain, okay, that's twisted. Those hydrogen bonds twist it, and it resembles a coiled helical spring, which is where we get the term alpha helix from. All right, so more on this alpha helix. All right, what causes this coiled spring type alpha helix? Well, as we said, this shape, right, is held like this by hydrogen bonding interactions. How did those arise? Between the amide groups, right, with the side chains extending outward from the coil. That's what we saw on the previous slide, right? So let's just show you what we're talking about here. So the amide group, right, on one with another one down further, right? The carbonyl down further. So we get that hydrogen bond, and that then gives it that distinct coil shape. The amount of alpha helix coiling in proteins is going to be highly variable depending upon who the groups are. Okay. Examples. We have alpha keratin, which is a protein found in hair, and highly variable, right? Some people have very straight hair, other people have very, very curly hair. Okay. Myosin in muscles, epiderm in skin, fibrin, which helps in the clotting of blood, right? These are all alpha helixes, and they all vary, right? Every single one of these is different, okay? So the amount of coiling is different, and that then results in a different function. In all of these that we just mentioned, 2 or more helices coil together, and when this happens, it's called supercoiling, and you get cables, all right? These cables make up bundles of fibers that strengthen the tissues in which you find them. All right, still talking about secondary structure. We have another type of secondary structure, the beta-pleated sheet. The beta-pleated sheet is entirely different from that alpha helix. In the beta-pleated sheet, several protein chains lie side by side, and they're held side by side by hydrogen bonds between the adjacent chains. So this is what we're talking about, all right? Again, here is a model. So here we see the carbons, right? The oxygens, the nitrogens, the R groups, etc. Okay, but we can make out we have a polypeptide chain of protein, right here. Another protein here lying side by side with the first, side by side. Okay, and what is holding them side by side? Well, hydrogen bonds. Okay, hydrogen bonds between the adjacent chains. All right, so here we see the carbonyl oxygen and a hydrogen bond between the hydrogen on another protein chain. Okay, and that is holding them side by side in what looks like almost a fan. All right, a pleated sheet. Okay, entirely different than that alpha helix. The beta pleated sheet is less common than the alpha helix. It's found extensively only in the protein of silk. So this figure here, all right, that I have on this particular screen shows both types of secondary structures in a single protein. Remember what we said a protein is, right? 50 or more amino acid residues joined together. So within that 50, okay, we see random structure going on here. All right, we come over here and we notice that we have some beta pleating going on, all right, between this section of our protein and this further section. Okay, and then over in this section here, we've got some alpha helix going on. So the hydrogen bonding is imparting the alpha helix on it, that coiled structure. Here, the hydrogen bonding is creating the beta pleated sheet, and this is what goes on in proteins. So in one single protein, you can have a number of different secondary structures. All right, still secondary structure. Okay, we also have the triple helix. In the triple helix, 3 polypeptide chains get woven together. Glycine, proline, hydroxyproline, and hydroxylysine. Hydrogen bonding occurs between the hydroxyl groups, and this then gives you a super, super strong structure. This is the type of secondary structure that's seen in collagen, connective tissue, skin, tendons, and cartilage. Okay, so we've seen the primary structure. That's simply the arrangement, the bonding of the individual amino acids, and then the way those are bonded imparts places where you can hydrogen bond, okay, which gives rise to your secondary structure, either your beta-pleated sheet or your alpha-helix, all right? And then we move on to the tertiary structure of proteins. The tertiary structure of a protein refers to the bending and the folding of the protein into specific three-dimensional shapes. These structures result from 4 types of interactions between the R side chains of the amino acid residues. Right? So remember, those R groups, they're different. Okay, so depending upon what they are, we're going to see different types of interactions. What are some of the interactions that we can see between our groups? Well, we could see disulfide bridges. Remember, we talked about disulfides in one of our organic chapters. Same sort of thing. We're seeing that between the R groups on a protein. Salt bridges, hydrogen bonds, and the fourth one is hydrophobic interactions. All right, so we have 2 groups that, or groups that are not polar. All right, so they're going to not be water soluble. So hydrophobic interactions. Okay. Okie doke. Let's look at these. Let's look and see what we're talking about. All right, so this is a good example showing the tertiary structure of proteins. All right, so let's review. So again, 50 or more amino acid residues all joined together with those peptide bonds, right? So that ribbon type, pink ribbon type structure is showing us the peptide backbone. Okay, all right, and periodically here we're showing, okay, this is cysteine, all right, this is asparagine, okay, and remember there's sulfurs, and so cysteine, all right, between 2 of them at different places on your protein, you can form a disulfide bridge between the thiols, all right, and here we're seeing a salt bridge. Salt bridge is going to form from a carboxylate group on one and the quaternary ammonium salt on another. We get a salt bridge, right? Alrighty, and here would be hydrophobic interactions, right, with benzene rings. Okay, they are definitely not polar. Okay, so hydrophobic interactions where you have 2 of them together, all right, because of the folding. And here would be a hydrogen bond. Okay, serine, right? 2 serine units, right, with their hydroxyl groups on them, perfectly set up. If the folding brings them in contact, right, they're going to hydrogen bond. All right. So that's the tertiary structure. It involves interactions between the side chains. Right. Okay. All right, let's see if we can visualize protein structure with everything that we have. All right. So we have the primary structure. The primary structure is simply the sequence of amino acids, the backbone. Okay. And the secondary structure then imparts, and so we're showing here the alpha helix, right? Secondary structure, depending upon what those R groups are on the chain, imparts the secondary structure. Okay, the coiling, all right, from the hydrogen bonding, that's one type of secondary structure. And then interactions between the R groups, the groups on attached to the R groups, impart the tertiary structure, right? The kinks, like we saw in the previous slides. So this cord here, where it's kinked together, it's held in this manner from interactions between the R groups. All right, that's the tertiary structure. How about the quaternary structure of proteins? When two or more polypeptide chains are held together by disulfide bridges, salt bridges, hydrogen bonding, or those hydrophobic interaction, what's going to happen? It's going to form a much larger protein complex. The one that we looked at a couple slides ago, that protein structure that was showing the hydrophobic interactions, what we meant, the hydrogen bonding between the R groups, the salt bridges, the disulfide bonds, right? That's tertiary structure, okay? All right. Now we're talking about one or more chains also exhibiting this. You're going to see a much larger complex, right? Each of the polypeptide subunits, right, the protein subunits, has its own primary, secondary, and tertiary structure. So if you can imagine, a couple slides ago, several of those, all right, each with their own primary, secondary, and tertiary structure. The arrangement then of these subunits to form a larger protein is known as the quaternary structure of the protein. Let's see if we can give you a visual, all right, or an example at least. Hemoglobin. Okay, hemoglobin would be an example of quaternary structure. All right, let's show it all to you. It's got all of these aspects, right? Okay, so we recognize from a couple slides ago. All right, we've got 4 joined together, 1, 2, 3, 4. All right, 4 proteins, each with their primary, secondary, and tertiary structures. Okay, and they are then all joined together through any of those 4 types of linkages. All right, so this is hemoglobin, and hemoglobin, of course, we know carries oxygen to the blood. It has 4 polypeptide chains, 4 protein chains, right? We said that term is used interchangeably with proteins, and we can see them. They're each color-coded. Again, each of those polypeptides, those proteins, has primary, secondary, tertiary structure imparted to it. And then each chain also has a heme group to bind oxygen. And we can see a heme group right here, with this one right there, okay, and right there. And that is an example of quaternary structure, okay? Each has its own properties, right, that it brought into the quaternary structure. All right, we move on to protein hydrolysis. And again, we're going to go back. We see a term that we've heard before multiple times in our study of organic chemistry, hydrolysis, right? Okay, you should be familiar with that. All right, and again, proteins, what are they? Right, they're composed of amides, right? They're polypeptides. Amides can be hydrolyzed under acidic or basic conditions. So that's all a protein is, right? It's a lot of amide bonds. The peptide bonds in proteins can also be broken down under acidic or basic conditions. And when we break those, we're going to break them, depending upon how many we break, right? First into smaller peptides, or we can keep breaking them all the way down into the amino acids. And that's all going to depend upon hydrolysis time, temperature, and pH. So here's what we're talking about, right? Remember, proteins, that's all they are. A lot of amide linkages. Amides can be hydrolyzed, right? So that means reacted with water, right? Along with a catalyst, right? Hydrogen right? H plus, right? Acid or base, acidic or basic conditions we saw, just like an amide. And depending upon how long we do this, right? If we don't do it for too long, we're going to just get smaller peptides, right? It's going to break it at some points into smaller peptides. And if we continue on, all right, with the hydrolysis, we could go all the way and break that protein down into its individual amino acids. And that's how they sequence proteins, all right? They just hydrolyze the heck out of them until they get all of the different amino acids and they figure out who's who, okay? So when proteins are digested in the body, that's all it is hydrolysis. The digestion of proteins simply involves hydrolysis reactions, except for in the body, those hydrolysis reactions are going to be catalyzed by enzymes, all right? In the lab, this would probably take heat to do it also, okay? In the body, those digestive enzymes help everything, reactions that may not occur at 98.6 degrees Fahrenheit. It helps them occur at 98.6 degrees Fahrenheit. Okay, so denaturation. Denaturation, we denature proteins oftentimes. What is denaturation of proteins? It is a disruption of the secondary, tertiary, and quaternary protein structure. Okay? And how do we do that? How can we denature a protein? We can use heat or organic solvents. If heat and organic solvents will break apart those hydrogen bonds that we see in the secondary, tertiary, quaternary protein structure, and it will disrupt any hydrophobic attractions that you might have in the quaternary structure. We can denature proteins using acids and bases. Again, acids and bases are going to break the hydrogen bonds between polar R groups. It's also going to break ionic bonds. Heavy metal ions will also denature proteins. Heavy metal ions react with those disulfide bonds and form solids. We can also just agitate a protein. Agitating a protein will stretch the chains, and if you stretch the chain enough, the bonds are going to break. Okay, the hydrogen bonds and any of these other interactions, right? So this particular slide, what I did was to put together some examples, all right, of the substance or the condition that you can use to denature proteins, and again, how it affects the protein, and some examples there too. For instance, all right, heat and ultraviolet light. All right, we've heard a whole lot about that, all right, with regard to the COVID-19 virus, right? Okay, that these are some of the things that kill the virus, which is made up of protein, all right? Heat and ultraviolet light, all right? What does it do? How does it kill and denature the protein in the virus? It disrupts the hydrogen bonds and the ionic attractions by making the molecules vibrate too violently, and it produces coagulation, such as cooking an egg, all right? But this is also why the virus is killed by heat and ultraviolet light. Organic solvents, okay, such as ethanol, right, and other miscible things that are miscible with water, all right? So ethanol, alcohol, hand sanitizer, all right? What is it doing? How is it killing bacteria and viruses? Well, it's denaturing the protein. You denature the protein, you kill it, all right? So again, what's it doing? It's affecting that secondary, tertiary, and quaternary structure. It's disrupting the hydrogen bonds in the proteins and probably forms new ones with the proteins so that it can then be washed away, okay? So in terms of the soaps, right? Strong acids and bases, again, disrupt hydrogen bonds and ionic attractions. Prolonged exposure with acids and bases. Remember what happens with acids and bases, right? And proteins, you're going to hydrolyze them, okay? You hydrolyze them, you're breaking them down, all right, so that they don't work anymore. Detergents, okay, disrupt hydrogen bonds and those hydrophobic interactions and the ionic interactions. Heavy metals, such as mercury, silver, lead, they specifically act on those thiol groups. Okay, so they form bonds to thiols, and they precipitate proteins as insoluble heavy metal salts. Okay. And I added in here something interesting because we're all concerned at this point with hoping that a vaccine becomes available. All right, so just a little piece of information here that interesting. All right, in light of the current situation, heat denaturization. Okay, up here. All right, is actually used to prepare vaccines against some diseases. So what did we say? That heat denatures the protein, right? The denatured toxin, all right, then can no longer cause whatever disease it is that they cause. But it can stimulate the body to produce antibodies for it, to produce substances that will help induce immunity. And that is one of the techniques of making a vaccine. There are many, but this is one that's used. They use denatured protein from the virus. All right, last topic, enzymes. Enzymes, what are they? They are catalysts for biological reactions. Catalysts, again, we have seen that term over and over and over. It is something that we add to a reaction to help it occur, okay? And the reaction will not occur without that catalyst. The catalyst is typically regenerated at the end of the reaction, okay? So all it's there is is to help the reaction go. The reaction will not go without it. And enzymes are there to help these reactions go in the body. The reactions won't go in the body without the enzyme. Most enzymes, okay, are proteins. Okay, so now we understand what a protein is. So here is what we just said. What is the role of an enzyme? Okay, it lowers the activation energy of the reactions, the amount of energy. This is one of the ways that it helps the reaction occur. It lowers the amount of energy that's required for the reaction to occur. And oftentimes, these enzymes increase the rate of reaction. Right? The reaction might take days to occur, but an enzyme's going to help it go within maybe a matter of hours, maybe a matter of minutes. Okay? Since enzymes are proteins, keep in mind that anything that applies to proteins applies to enzymes. So the activity of an enzyme is going to be lost if the protein from which it's made is denatured. Enzymes could be very simple proteins, or they could be a little bit more complex. They might contain cofactors, such as metal ions, or organics, right, such as vitamins. So we haven't looked at nomenclature with too many of these things, but we're going to look at names of enzymes here because they're pretty interesting. Enzymes. How do you know that you're dealing with an enzyme? All right. Well, enzyme names always end in ASE. So that's a hint. ASE ending, it's an enzyme we're talking about. And the enzyme name identifies oftentimes the reacting substance, the thing that they're catalyzing a reaction for. For instance, sucrase. Okay, the ASE ending enzyme sucrase, all right, that prefix, it's reacting with sucrose. All right, so the name often identifies the reacting substance. Lipase, all right, an enzyme that reacts with lipids. The name also, all right, sometimes describes the function of the enzyme. Oxidase, again, the ending tells us we're dealing with an enzyme, all right? This one, the prefix is not telling us the reacting substance, but it's telling us the function, all right? What does this enzyme do? All right, well, it catalyzes oxidation, okay, oxidase. Hydrolase, right? It's an enzyme that catalyzes hydrolysis. Common names of digestion enzymes, however, are still used, okay? So be aware that some enzyme names are not going to end in ASE, and some examples, pepsin and trypsin, okay, no ASE ending, all right, but these are digestive enzymes. Okay, so different levels. These are very specific names, all right, because they're telling you the reacting substance. These are a little bit less specific. They are indicating the function. Okay, so the naming has several levels to it, so to speak. All right, more on the names, classification of enzymes. All right, so let's look at some classes of enzymes. They're classified according to the reactions that they catalyze. So, oxidoreductases, all right, is a class of enzymes. And the reactions, of course, that they catalyze are oxidation and reduction. You can get that very easily from the name. Transferases. What reaction do they catalyze? Well, the transfer of groups of atoms. Hydrolysis. This is an easy one, right? Reaction that they catalyze is hydrolysis. The lysases or lyases add or remove atoms to form a double bond. Lyases. Isomerases, right? Again, we see that ASE ending, right? So we know that these are all enzymes. What's the reaction that they catalyze? An isomeration reaction. What's isomeration, right? Well, we're going to rearrange atoms and form isomers. Ligases or ligases, all right, combine molecules using ATP. Let's give you some examples of enzyme classification, all right? So let's remember the class, the oxidoreductases. They catalyze oxidation reduction reactions. Under this general class, we find the oxidases and the reductases, all right? Oxidases, of course, oxidize, and reductases are going to reduce. Transferases. Under the transferases, that general category, we find the transaminases, okay? These transfer amino groups. The kinases, they transfer phosphate groups. In that main category of hydrolysis, which catalyze hydrolysis, we have the proteases. They hydrolyze peptide bonds. The lipases, on the other hand, they hydrolyze lipid ester bonds. The lyases, in this category, we have the carboxylases, which add carbon dioxide, and the hydrolases that add water. All right, we're nearing the end here. One of the last categories of discussion regarding enzymes. Let's look at enzyme action, how they work. There are a couple different models right, for enzyme action. The first model is what's known as the lock and key model. In the lock and key model, an enzyme binds a substrate in a region called the active site. Now, only certain substances can fit the active site. In the lock and key model, it's like a lock and a key. All right, you have a lock on your door of your house, right? Okay, and only certain keys will work in that lock. So it's the same sort of premise with the enzyme. Okay, only certain substances can fit into that active site. Amino acid R groups in the active site are what help the substrate bind. Right? Depending upon the R group, you can get certain interactions. And when you get this interaction, you're going to see an enzyme substrate complex form. Once this forms, you can get reaction because the things that need to react are close enough. The substrate reacts and it forms a product. And the product is then released. So here it is. Here's this model, okay, the lock and key model. We see here, beginning at the far left, we see the enzyme, okay? It's like our door lock, okay? There it is, right? If we could peer inside our door lock, it has a certain configuration, right? The substrate, right? The substrate has a configuration, right, that fits appropriately into that lock. All right, so it's the key. It's got to fit in there. If it fits, right, if it has the appropriate configuration, we get the enzyme substrate complex forming and the reaction occurs. Okay, they're held tightly together so the reaction can occur. And once the reaction occurs and product is formed, the product is released. And we're back. We have the enzyme back and it can operate again. Okay, so that is the lock and key model. But that's not the only model, all right? There's yet another enzyme action model, right? So this is the induced fit model of enzyme action. In this model, they believe that the enzyme structure is not rigid as it was in the lock and key, okay? That it's more flexible. The enzyme then and the active site can adjust the shape in order to bind the substrate. And this then increases range of substrate specificity. The shape changes also, and this improves catalysis during the reaction. So let's look and see a pictorial of what we're talking about here in this induced fit model. All right, we still have, as we did before, the enzyme. Okay, so here on the far left, this is our enzyme. And if we look at it, it doesn't look like it's going to fit together, right, with our substrate here. Okay, definitely it's not the puzzle piece, all right, but it's flexible, as I said, both of them. So even though it doesn't look like it fits, the flexibility that it has, right, to adapt its shape so that, yes, you can form this enzyme substrate complex. Okay, and again, in this enzyme substrate complex, the enzyme, the substrate are held together so that the reaction can occur. Once reaction occurs, all right, the products are released and our flexible enzyme, all right, is back and ready to go again. All right, so those are the two models of enzyme action. All right. So in both of these models, there are factors that do affect enzyme action. And we're going to go through those one by one. One is temperature. At low temperatures, very little enzyme activity occurs. You have to have optimum temperature for enzyme activity. As you increase the temperature, the rate of enzyme reaction increases. Most enzymes have an optimum temperature for the reaction, and it is usually 37 degrees Celsius in humans. So that's 98.6 degrees Fahrenheit. And too high a temperature, and the activity of the enzyme is lost. And remember what happens, all right? At too high temperatures, at high temperatures, we can denature proteins. Enzymes, what are they? They're made of proteins. So it's not surprising that at high temperatures, really high temperatures, they're going to lose their activity. And again, it's because of denaturation of the protein. Substrate concentration is another factor that affects enzyme action. If you increase the concentration of the substrate, that is going to increase the rate of reaction, right? And when we say we're increasing concentration, keep in mind the enzyme, its concentration, we're leaving that the same, all right? That's constant. So the only thing we're doing is we're increasing the substrate concentration, and that, yes, increases the rate of reaction of the enzyme. Maximum enzyme activity is reached when every bit of your enzyme combines with a substrate, okay? You're going to get maximum amount of product at that point. All right, what else affects enzyme action? pH. Maximum activity of an enzyme occurs at optimum pH. The R groups of the amino acids have to have the proper charge in order to have that optimum pH. The tertiary structure of the enzyme has to be correct. And there is a very narrow range of activity when we're talking about the pH. Most enzymes are going to lose their activity if the conditions are too acidic or too basic. And your textbook in this chapter, they talk about some of the conditions that can happen if the blood pH ends up too high or too low. Remember, the blood pH is about 7.4, okay? Just ever so slightly basic, okay? Too low or too high, and your enzymes then do not work, and that leads to a cascade of effects within the body. Enzyme inhibition. Inhibitors are going to cause a loss of catalytic activity. What inhibitors do is they change the protein structure of the enzyme. Inhibitors can be one of two types. They can be competitive inhibitors, or they can be non-competitive inhibitors. Some enzyme inhibition are reversible. Some are irreversible. Let's look at these two types of enzyme inhibition. The competitive inhibition first. A competitive inhibitor, what do we mean by that? It is going to have a structure similar to the substrate. Okay, so protein structure similar to the substrate. So that means it's going to be able to fool, right, it. And it's going to occupy the active site. And it will then compete with substrate for the active site. It has its effect reversed by increasing the substrate concentration. Okay, so if you increase the substrate concentration, you can in fact reverse the effect of a competitive inhibitor. And then our second type of inhibition, enzyme inhibition, is the non-competitive inhibition. A non-competitive inhibitor is not going to have a structure like the substrate. So it's going to bind to the enzyme, but not that active site where the reaction occurs. It's going to change the shape of the enzyme and the active site. This is how they operate, a non-competitive inhibitor. And when you change the shape of the enzyme, remember it was dependent upon that shape, all right? So we change the shape, and that substrate then cannot fit into the changed active site. So it's disrupting the whole reaction. No reaction is going to occur then, and then you get no products. So the effect of a non-competitive inhibitor is not going to be reversed by adding the substrate, right? Any more substrate, because it doesn't matter how much you have, it's not going to fit that changed active site in the enzyme. And that's it, folks. All right. We have gone through every single topic on our syllabus, and next week it is exam 4 time. So begin that studying